In today's video, we're going to go over immunity for AS level biology. By the end of the video, you're going to know the processes involved in the first and second lines of defense. So without any further ado, let's jump into the first line of defense. The first line of defense is non-specific to a certain pathogen. It will work against a variety of different germs, bacteria, or pathogens in general in the same way. Now, what is a pathogen? So a pathogen is a microorganism that causes disease. So now let's take a look at some of the barriers that we have in our first line of defense that prevent the entry of these pathogens into our system. One of the first barriers is a physical barrier, which is skin, right? This prevents the entry of pathogens into our system. A chemical barrier is the fact that in our tears and in our saliva, we have an enzyme called lysozyme, which causes the lysis of bacteria. In our stomachs, we have hydrochloric acid. So if a bacteria is to enter into our stomachs, that hydrochloric acid will provide a very low pH environment, which will cause the denaturing of most bacterial enzymes and then the subsequent death. We also have epithelia that are covered in mucus. So for example, in our tracheas, we have ciliated epithelium. So goblet cells will secrete mucus, which will trap pathogens, and the cilia will waft those mucus-trapped pathogens away from the alveoli and up to the pharynx. Now, it's important to understand that pathogens can bypass all of the barriers that we have in the first line of defense. And in that situation, the second line of defense is then called on. Now, it's important to understand the concept of self versus non-self antigens. So the concept of self versus non-self refers to the idea of whether or not an antigen stimulates an immune response. If an antigen does not stimulate an immune response, it is a self-antigen. If it does stimulate an immune response, it's non-self. So make sure that you don't get confused with the fact that self means it's mine and non-self means it's the outside environment. No, it simply refers to the idea of whether or not the antigen in discussion stimulates an immune response. Now, let's take a look at white blood cells. So we have two types. We have phagocytes and we have lymphocytes, which are involved in the second line of defense. Now, before we go on to the types of phagocytes, let's quickly take a look at what phagocytosis is. So phagocytosis is the uptake of one cell, so the uptake of a larger particle by a cell. And the process by which this occurs is that the cell surface membrane of the phagocyte forms these arms and it wraps around the pathogen to engulf it. It's important also to understand that as a result of phagocytosis and the formation of a phagosome, which is a vesicle containing the outside particle, the cell surface membrane of that phagosome, of that phagocyte, does in fact become slightly smaller. Now let's take a look at the process of phagocytosis with a neutrophil. So a pathogen, a bacteria, enters our system. Complement proteins from within our blood plasma will attach to that bacteria. And the bacteria may also be releasing chemical products. And these two factors will attract the neutrophil towards the bacteria to stimulate phagocytosis. And because they stimulate phagocytosis, they're called opsonins. So the neutrophil will reach the bacteria, and then with its receptors, it will bind to the opsonins, and then it will engulf the bacteria. The bacteria will then be in a vesicle within the phagocyte, and that vesicle is called a phagosome. Lysosomes from within the neutrophil with their hydrolytic enzymes will fuse to the phagosome and release their hydrolytic enzymes into the phagosome, which will then go ahead and break down the bacteria into soluble material. Now, the soluble material will be absorbed by the cytoplasm of the phagocyte, and waste material will be released out of the phagocyte. Now, monocytes can do some phagocytosis and they can also present some antigens. But what monocytes are really good at is that they can migrate to tissues that need white blood cells, and then they can differentiate into macrophages, which are very efficient at phagocytosis, and they also do a lot of antigen presenting. So these macrophages, the way that they will present antigens is that they will break down the bacteria or pathogen, as we discussed in the process of phagocytosis, and then they will present those certain proteins or antigens on their cell surface membrane for T lymphocytes to come and be stimulated by in the second line of defense. Now, before moving on to the second line of defense, it's important to recognize, so neutrophils can easily be identified because of the fact that they have a very irregularly lobe-shaped nucleus, whereas monocytes have a more regularly kidney-shaped nucleus. Now, as we discussed, monocytes will differentiate into macrophages. So the neutrophils will be the primary responders. They will be the first types of phagocytes to respond to a foreign pathogen, and then later on, the more efficient macrophages will then be able to also act and do phagocytosis. So as we move on to the second line of defense, the second line of defense is specific to certain antigens. Okay, certain antigens on a pathogen will stimulate a B lymphocyte or a T lymphocyte. Now, what is an antigen? So an antigen can either be, it's generally a protein, however, sometimes it can be a glycoprotein, so that means that a carbohydrate is attached to a protein, and other times it can also be a polysaccharide. So if we take a look at how T lymphocytes will be activated by an antigen presenting cell, let's take a look at the T helper cell. So the T helper cell will attach to the antigen that is being presented by an antigen presenting cell. Now the T helper cell will then divide by mitosis into more T helper cells and memory T cells. Okay, now what T helper cells will also do is that they will release cytokines, which will stimulate T killer cells and B lymphocytes, and T helper cells will also be able to recognize the antigen and then go directly to the B lymphocyte to stimulate the B lymphocyte, to activate the B lymphocyte. Before we get to B lymphocytes, let's look at T killer cells. So T killer cells will attach to the antigen on an antigen presenting cell, and they will become activated. Now, on top of the fact that the cytokines from the T helper cells stimulate the mitosis of T killer cells, the T killer cells will also naturally just divide into more T killer cells and also memory T killer cells for that specific antigen. And the way that T killer cells work is that they bind to a cell that is presenting the certain antigen, and then they release perforins, which go ahead and make holes in the cell surface membrane of that cell, allowing water to enter the cell, causing the bursting and death of that infected cell. Now, memory T cells we'll get back to when we reach the secondary immune response. But if you've been able to notice, T lymphocytes deal with cells that have already been infected and are presenting antigens. Now, on the contrary, B lymphocytes interact with antigens that are floating within the system. Okay, so it's very important to understand T lymphocytes deal with the antigen with cells that are presenting antigens, whereas B lymphocytes will interact with the antigens that are floating within the bloodstream. So B lymphocytes, they can become activated by T helper cells, and then what also happens is B lymphocytes can bind directly to the antigens, as we've just discussed. Now that will activate them, and then they will grow, and then they will do a number of mitotic cell divisions to produce many of the same types of B lymphocytes in a process called clonal expansion. And this clonal expansion is also stimulated by the cytokines that the T helper cells released. And then, after we have many clones of that B lymphocyte, those B lymphocytes will develop into either plasma cells or memory B cells, which we'll get back to when we reach the secondary immune response. So plasma cells produce antibodies, right? Up to about 2,000 antibodies per second. And so therefore they need to produce a lot of protein. Now they're very well equipped for this because, number one, They have a large nucleus for a lot of mRNA transcription, right? For a lot of mRNA synthesis, for a lot of transcription to occur. They also have extensive rough endoplasmic reticulum for a lot of polypeptide production, and also extensive Golgi body for further processing of those proteins. Now, this is all the primary immune response. Okay, so the primary immune response has been our T helper cells, our B lymphocytes are becoming activated, they're producing more B cells, plasma cells, memory B and T cells, etc. That's the primary immune response. Now, after we have the primary immune response, we have memory B and T cells. Now, these are going to provide long-term immunity. These memory B and T cells will remain in the system for many years. And then the next time that we become infected with the same pathogen that has the same antigens, these memory B and T cells will be able to act on that pathogen very quickly and prevent the growth of that pathogen to cause disease in the human again. Okay, so those memory B and T cells will encounter the pathogen very quickly, and then they will be able to produce, so the memory B cells will be able to quickly produce more plasma cells, which will then release antibody, and then the pathogen will be very quickly destroyed. And then likewise, T killer cells will destroy the pathogen directly. So the secondary immune response happens much faster than the primary immune response, and it's also much more powerful. Okay, because we have those memory B and T cells already present within the bloodstream, they very quickly encounter the pathogen and they very quickly act to kill it and eliminate it. So that's it for this video. If you found it helpful, I'm very glad. Take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thank you very much for watching, and I hope you have a great day. Bye-bye.